I think this is 6.01. So this is about the classification of living things, right?
Mm-hmm.
So you've probably heard of like, like things like kingdom, phylum, class,
Phylum, species, class, yes, all
right? All
that.
those kinds of things, right? So we're going to go over, that classification system today. So, this picture over here, right? This picture over here is something that that early scientists or early philosophers came up with. So there was a Greek philosopher named Aristotle. So he recognized the need for a classification system for living things. So he came up with this, this sort of classification system where there was like kind of like this hierarchy, right? Like from the least
Mm-hmm.
complex to like the most complex. So for example, like plants are below animals, which are below humans, and then within animals, right, it's like divided up, right? For example, like more like mammals are at the top, and then kind of invertebrates and that kind of thing are
Mm-hmm.
near the bottom. So this is kind of like a classification system that that this person came up with, like
Mm-hmm.
in terms of. So this was organized in terms of like complexity, right? Not necessarily in
What do
terms
you
of
mean in
like,
terms of complexity?
so like the higher ones, the ones at the very top are more complex. The ones at the bottom are
Oh, okay. I
considered
see
to be
what you're
less
saying.
complex,
Less
right?
complex. I see what you're saying.
Right. So this did not include anything to do with like Darwin's theories of evolution or anything like that. This was
Mm-hmm.
simply less complex to more complex.
Mm-hmm.
All right. So what we are more familiar with is something called is these kinds of classifications. So there's a person named Linnaeus, right? So he divided nature
Mm-hmm.
into three kingdoms: animals, plants, and minerals. So minerals are not actually living things, right? They're actually non-living things like rocks and
Mm-hmm.
soil. But he just he divide he divided nature into these three portions: animals, plants, and
And
minerals.
minerals.
So this also was not a result of like some kind of theory of evolution. This was just based on comparing physical characteristics and then
Mm-hmm.
saying that these creatures are more alike, so we're going to group them together. These plants are more alike, so we're going to group them together like this.
Mm-hmm.
So this kind of classification system still did not include any of Charles Darwin's ideas. Right.
Mm-hmm.
So this right over here, right, is an example of how we classify, species, different kinds of species today. So every creature, belongs to one of these groups, right? So you have domain, kingdom, phylum, class, order, family, genus, species, right? So species is obviously like the most specific one, right? Right. Species is the most
But
specific.
but oh, okay. So the one on the left is like from bottom to top, right? The
Yeah,
same thing, but this from
yeah,
bottom to
yeah,
top.
yeah. This is the same thing, just in a different
Uh,
example. So this over here is given.
domain.
Mhm.
Domain is. We're gonna talk about each one of these, like what it represents, right?
Right.
Okay.
Kind of in some detail, not necessarily, but in some detail.
Don't. But this one doesn't have domain. This one has domain. Domain is just if it's eukaryotic or prokaryotic.
Right. So there's three domains. One is called bacteria, one is
Mm-hmm.
called archaea, and one is called
Mm-hmm.
eukarya. So bacteria and
Okay.
archaea are both prokaryotes, and then
Prokaryotes. All
and
right.
then the other one is eukarya, which is eukaryotes. Right. So
Mm-hmm.
basically, within each genus, there are multiple species. Within each family, there are multiple genus. Within each order, there's multiple families, and so on. Right.
Mm-hmm.
So this is just giving you an example for this fox, which
Yeah.
domain, kingdom, and so on that it belongs to.
Mm-hmm.
Now, this one over here is giving, one way of organizing different things. So over here, so in terms of, so when we looked over here at Linnaeus's kingdoms, right, there were three, right? Animals, plants, and minerals. So these were the kingdoms that this person came up with. So today there's considered to be 5 different kingdoms. Right? So remember we have bacteria. So in terms of domains, we have 3: bacteria, archaea, and eukarya. Now, in terms of kingdoms, there's considered to be 5 today. So when it comes to bacteria, within that there's just one kingdom, eubacteria. Right? Within archaea, there's just one kingdom, archaebacteria. Right? Does that make sense? So usually within each classification group, like if you go down, there's then it branches out, right?
So this prokaryotic thing is a domain, right? Where it says prokaryote, and then inside that is archaebacteria and eubacteria, both of which are kingdoms.
Not quite. So I think this is from the what do you call it? From the FLVS. So I just put in here because it was from the thing, but I don't think this is the best picture because it is a bit confusing, right? So prokaryote is not really a domain, right? So these are not 2 domains. So if you want this, so I think these are better pictures. I don't think that picture is like the best. So these are the 3. These are the 3 domains. Bacteria, archaea, and eukarya. These are the 3 domains, right? So this picture over here, I think, is just showing that these 2 kingdoms are prokaryotic kingdoms.
Oh, okay. And then eukaryotic are all these other kingdoms.
Right. So today there's a total of 5 kingdoms. Eubacteria, which is basically
plantae, fungi, animal. Wait, it says kingdom Protista, for example. Is that a kingdom?
Right, that's a kingdom.
And then we have plantae, and then we have fungi, animalia. And we have one more. Where's the last one?
So I think archaea is also. So I don't think this is correct. So it says currently the 5 kingdoms are. So I think there's actually 6, right? So there's eubacteria, which is like bacteria, right? Archaebacteria, which is archaea. So first of all, what is the difference between like bacteria and archaea? Do you know what is the difference between them?
Bacteria and archaea? No, I don't.
So they're both prokaryotes, right? But the difference is there's some differences in their cell wall structure. So so bacteria is like what you normally think about. It's like what people get sick from and that kind of thing. And then archaea is also prokaryote, but it basically lives in mostly like extreme conditions. So for example, like
it's basically bacteria, but it's like lives in extremely. All right, I see what you're saying.
So you can think of it that way. So there's also differences in the cell wall. There's some differences in the structure, but they're all prokaryotes. So they're all pretty simple kinds of organisms. But archaea lives in extreme conditions, like very salty places, high temperature places, very acidic places, things like that. Like very extreme places. That's where archaea lives.
All right, I see.
And then bacteria just lives anywhere, like just all over the place. All right. So these are the 2 prokaryotic kingdoms, and then we have 4 eukaryotic kingdoms, right? Animalia, fungi, plantae, and protista.
So there's 4 prokaryotic kingdoms?
4 eukaryotic kingdoms.
And then how many prokaryotic?
2.
All right, 2. So archaebacteria and eubacteria.
Right.
All right.
So these are the 3 domains, right? This is where it starts: bacteria, archaea, and eukarya, right? So within bacteria and archaea, there's only 1 kingdom each, right? But then there's 4 in eukarya. Does that make sense? All right. So so these are the 4. So down here you have the archaebacteria, eubacteria. These are the prokaryotic ones, and these other 4 are the eukaryotic ones, more complex cells. All right. All right. So when we when we classify a creature, right? When someone gives like the name of a creature, like, what is the scientific name of this creature? What you see is you usually see 2 different names, right? Like Homo sapiens, right? So where did those 2 names come from, right? Homo sapiens, like what is that referring to? Where are we getting 2 names from?
Homo means like what's it called? Same, and then sapiens. I'm not sure.
Um, I'm not sure about the. I don't mean like this. Where did these specific things come from? But how come there's two names, right? So what are the
What do you mean?
two?
How come there's two?
So where are the two names coming from? The
Oh,
two
why
names,
is it like Homo sapiens? Is what you're saying?
right? So usually the scientific name includes the genus and the species, right?
Oh, okay.
Right?
Oh yeah, okay, yeah. Homo and then Homo sapiens. All right, I
Right.
see.
So that's called this is called
Hominid.
this is called binomial nomenclature. So genus
Mhm.
plus the species. That's how you normally refer to a species. So family and everything above, right? All these other things, you don't
Mhm.
usually cite. You just cite the genus
Mhm.
and the species.
All right, I see.
All right. So this system of naming is called binomial nomenclature.
Nomenclature.
Right.
All right.
Right. And this is like these names they usually come from Latin. Right.
Mhm.
All right. And this is just giving some more examples. So here's the pebble crab, genus and species. So together, you give it that name. Lion, again, its genus is Panthera, its species is Leo. So together, Panthera leo, all together.
I see.
Sorry, what happened? All right.
Can
So
you go back to like the previous picture?
this one.
The lion one, yeah. All right, all right. See, so this the scientific name for a lion is Panthera leo.
Mm-hmm. So
All right.
right.
Okay, so I see how it works.
Within this genus there could be multiple species. But so you put the genus and species together and you get
Yeah.
its scientific name.
Mhm.
All right. And then this over here, right, it's just giving you like some examples. So within this order, right, Carnivora, there are multiple families, right? And then within the family, there are usually multiple genus. And then within the genus, there's usually multiple species.
There's a lot of genuses, right, and like families and stuff.
Right. Hmm. So, for example,
Species are
within this
probably a lot.
genus, you have two
Mhm.
different kinds of cats.
Oh.
Right. Within this genus, right, there's two different kinds of bears.
Mhm.
Right. This is also another kind of picture you might see more often, like in scientific journals and stuff like that. So within this order, right, you see how it branches off into these multiple families. This family,
Yeah.
you can find class, order, family, genus. So within this family, there's multiple genuses, and then within this
Mhm.
genus, there could be multiple species. Right.
Mhm.
So before, like historically, right, like how do they determine how to group organisms together, like within the same genus or within the same family, order, stuff like that? What they did was they compared their physical
Physical traits, right?
characteristics, right?
Mhm.
Their physical characteristics, right? So before they didn't have access to like the looking at the DNA sequences and that kind of thing, proteins. So they would just kind of say, okay, well these two creatures they have like 12 things together, whereas
Mhm.
these two creatures only have like five. Maybe you have an animal, right? And you're comparing
Mhm.
it to two different creatures. So you say, okay, with
Mhm.
one, this creature has like 12 similar characteristics. But with the
Mhm.
other, there's only like maybe five. So okay, so we're
Mhm.
gonna put the two that have the most characteristics together in the same genus,
Mhm.
something like that.
Yeah.
So, um, so that may or may not be correct. So with the so along with those physical characteristics, you could also look at DNA sequences, protein sequences,
Mhm.
things like
Yeah.
that, and then compare and say, okay, well these two have the most similar amount of DNA, and so we're going to group them
Mhm.
together, like that, right? So there's both physical characteristics, and you could use genetic characteristics or chemical characteristics, right?
Uh huh.
So that's another way that people are now trying to figure out how to group things together, right?
No.
So in the 1970s, scientists began to use nucleic acid sequences and ribosomal RNA
RNA.
to determine links between organisms. Right?
Mm-hmm.
So they're using physical characteristics and RNA. So they're saying that based on the RNA sequences, they figured out that eubacteria and archaebacteria are different kinds of things. So before that, they just thought, oh, they're all prokaryotes, they're all kind of the same.
Mm-hmm.
But then they noticed, okay, there's two distinct, very different, distinct kind of groups. So maybe it's better to put them separately into separate domains. Right?
Mm-hmm.
So this is how they got bacteria and archaea separately, by looking down at the RNA. Because maybe on the surface, if you look at a microscope, through a microscope,
Mm-hmm.
you may or may not see necessarily too many differences. All right, this is just kind of telling you some similarities and differences between these different groups. So you notice that archaea and bacteria, right, they have some similarities. So they're both prokaryotes. So they lack a nuclear envelope, they lack membrane-bound organelles, right? But archaea and bacteria, see at the top, there's a difference with their cell walls, right? So
membrane lipids with branched hydrocarbons.
Right. So with their cell walls and cell membranes, there are differences, right?
Uh-huh.
And of course, eukaryotes are don't have any of these characteristics, because they're not prokaryotes. Right. So this is just kind of showing you some different similarities and differences between these different kingdoms.
Yeah.
All right. So that's one way of how people generally used to classify organisms before is by looking at their characteristics, kind of like counting the number of similarities and differences, right? So now what people are trying to do is trying to classify, trying to figure out, based on the theory of evolution, right? At every point, like let's suppose you take this graph over here, right? So at every junction, right, at every fork in the road, basically, from one common ancestor, the organism was split off into two different groups, right? So for example, right over here, let's say right over here, right? So at this branching point, some organisms started going down this way, and that ended up becoming bacteria. Some organisms started going down this way, and that ended up becoming archaea and eukarya. So the idea is that, from counting characteristics, people are shifting their focus to trying to see, are these creatures, which creatures, had a common ancestor. So it's kind of sort of the same thing, right? If two creatures have a common ancestor, more likely than not, they're going to have similar physical and chemical characteristics. Right? So it's kind of sort of still the same kind of thing, but you see how it's just kind of a difference in like the perspective that you're looking at the things. Right?
Yeah.
So the idea is that, okay, so still we're looking at similarities because we assume that, okay, if two things have, similar characteristics, then they probably had a common ancestor. But not necessarily the case. Maybe, once something split off, it just suddenly evolved like very, very, very quickly into something else. So it might be very different from everything else now, but it's still closely related from the evolution point of view.
Yeah.
All right. Does that kind of make sense?
Yeah, it makes sense.
Okay.
All right, brother. Can you I need to make a very important call to my teacher because
All right. So we're talking about phylogeny, right? So here's the idea. Let me see. Where do we go? All right. So we're gonna talk about phylogeny and cladistics. So phylogeny is the evolutionary history of a group of genetically related species. So the evolution of species is represented by a tree of ancestors and descendants. So let's look at some examples. All right. So this thing over here is called a phylogenetic tree. All right. So here's what's going on, right? So we're imagining that okay, we're starting down here like a long time ago, and then as we go on this tree, at here it forks, right? Like some of the creatures they go over here and become the jawless fish, and everybody else continues down this way, right? And we keep going, and at each branching point, right, it splits off two ways. So this is basically called a phylogen. This is called the study of phylogenetics. We're studying how these.
What exactly does phylogenetics mean?
So it's basically just the study of, the evolutionary history of some species.
Genetically related species, or just any species, just the phylogenetic?
Well, I guess any species, but how they're genetically related together.
Oh, okay, I see what you're saying.
So in this case, right, at every branching point, we're assuming that there's, one group goes down one way, and the rest of the groups go down the other way, right?
Yeah.
Now within this, right, let's suppose we take, this comment. Let's suppose we're right over here, right, where it says amniotic egg, right? So we're right here on, right below this branching point, right? Like immediately below where it branches. So if we take this common ancestor right over here, right, then do you see how, all of these creatures, from mammals all the way to birds, all of these creatures, their common ancestor existed like right over here, according to this tree, right? So so if you take, the common ancestor and everything that results from it, right? Like this this kind of like kind of right triangle right over here, right? This right over here is called a clade, CLADE.
Clade, all right.
Right. So CLADE. So if you go over here, any place, right? What if I go over here, right here, all right? So this organism right here, and it's eventually became crocodiles and birds. So this common ancestor and everything that results from it is called a clade.
Wait, which organism? The organism that's like down there next to the vertebrae thing, or?
Do you see the mouse on the screen?
Yeah, I see the mouse.
Right. So there's like a creature right over here. All right. And
I don't see any creature right here, brother.
No, I'm just saying, like, so these are the creatures today, right? But we're assuming that they came from creatures in the past. Right. So all along these lines are like the ancestors of the crocodiles and the birds.
Okay, I see.
All right. So like this, so right over here.
So all of these, the turtles, mammals, amphibians, bony fish, these are all ancestors of the crocodile, or are we talking about animals?
Right. So these ones right over here. See the ones at the top? All of these ones at the top?
Yeah.
These are like modern creatures.
Okay. Yeah.
And then anything like, so they're at the ends, right? You see how they're at the ends of each line, right?
Yes.
But if you go down, back down the line.
Yeah, but why are they showing traits at the bottom then? If they're supposed to be animals, why are they showing traits?
Okay, yeah, no. So
amniotic egg.
Yeah, so we're going to talk about that. So, first of all, do you get the idea that along, where all these lines are, right? You could assume that there's like historical creatures that used to live.
Yeah. Like on each line, showing like kind of like the family tree, for example, in a way.
Yeah. So if we take this bird, and we go all the way back to a point where, there was a common ancestor with the crocodiles and the birds. Before they split off into two groups, there was some creature that existed here, right? So if we include that creature, and everything along this line until birds, and everything along this line until crocodiles, that's basically that common ancestor and all of its descendants, right? So if we take the common ancestor and everything that descends from it, that the term for that is called clade, CLADE. Right.
The common ancestor and everything that descends from it. All right.
Right. Okay. All right. Now you're asking about what are all these things doing over here? So these things are these things represent the like when each of these characteristics like came into being. Okay, so for example, let's take jaws.
Oh, okay. So all of these characteristics represent for the crocodiles and birds, right? And the hair, for example, is for the mammals.
Right.
So all of these amniotic egg, lungs, forelimbs, bony skeleton, jaws, vertebrae, all for the crocodiles and the birds.
Right. So for example, let's take this one over here. It says jaws, right? It says jaws. So right over here, there's a common ancestor that's approaching this branching point right over here, right?
Yeah.
And then so some of the creatures, they left and they're going down this way before they evolved jaws. So
okay, I see what you're saying. Like that's why it says jawless fish here.
Right.
Okay,
So
it's a vertebrae.
Right. So jawless fish, they do not have jaws, but you see how everything else.
Okay, I see. No.
Everything else occurred after they have jaws.
Right? Like cartilaginous fish.
Right.
Have jaws with like sharks, and then bony fish after the bony skeleton came, and then lungs and forelimbs for amphibians, and then amniotic egg. And we got hair for the mammals.
Right. Right. So
I see what's going on here.
Yeah. So basically, like once you pass each of these red lines, then everything after it has that characteristic. Everything before it does not have it.
So wait, hold up. So jawless fish don't have lungs? Oh wait, no, it's a fish. Never mind. They have gills.
Right. Yeah. So lungs evolved here. So everything before there, such as bony fish, cartilaginous fish, jawless fish, they do not have lungs. That's what it means. Right. So the study of all of this is called cladistics. Right.
Oh, I see what you're saying.
So cladistics is depicts hypotheses about how organisms are related based on the traits of their ancestors and descendants. So we're looking at each of these traits and trying to arrange them based on when these traits occur. So this whole study is called cladistics.
Okay,
Right?
brother.
Now this over here, this is same kind of thing, except they're not looking at. Remember, we're talking about physical characteristics versus chemical characteristics. So in this case, they're trying to make a similar kind of tree. So this is obviously like a lot more complicated, right? This is going to be more complicated to discuss. But in this case,
I can tell. It looks much more complicated. Yeah.
So they're trying to do kind of the same kind of thing as this, but looking.
You're lagging, brother. Hold up here.
All right. So can you hear me?
Yes.
Okay. Okay. Yeah. Okay. You were lagging.
Okay,
You're good now. Yeah, I can hear you.
So they're trying to do the same thing as this, except they're looking at the sequences for this gene. So now they're looking at the DNA sequence of cytochrome C, and based on that, they're constructing this more complicated tree. All right, but it's the same kind of idea. So you can use the physical characteristics or genetic characteristics. All right, and this is a similar one. It's basically the same kind of thing.
But wait, brother. But like the phylogeny tree, right? The phylogeny, whatever tree, the colorful one, slide 15. Yeah, that one. Is it like those? So those numbers are showing something that has to do with like their genetic makeup, right?
Right. So it says values are estimates of the minimum number of nucleotide substitutions that have occurred along the lineages in the gene coding for this protein.
So they're looking at like mutations or something like that, nucleotide substitutions.
Yeah.
Like sort of like.
Right.
So isn't that like sort of a mutation? Not necessarily a bad one, but like.
Right. Correct. So this is like a mutation. So presumably, either kind of like neutral, or maybe beneficial, for that organism.
Yes, something like happened in their genetic makeup that like changed something slightly, and then like that just kept happening eventually. Right? That's what's going on here?
Right. So they're trying to look at similarities in the sequences and grouping those that have something similar together. Right. This is the same kind of thing. They're just this is just the example in the book that they gave, or in the FLVS that they gave, but it's the same kind of thing, right? So over here, after hair evolved, so everything before this, like everything that branched off here doesn't have hair. But everything after this point, like all these guys over here, monotrines to placental mammals, they have hair. So this is the same kind of thing.
Mm-hmm.
So the idea is that the simplest organization of evolutionary connections is usually the best arrangement. So here's what that means, right? Let's suppose,
Mm-hmm.
all right, so this over here, it presumes that hair evolved one time, right?
Mm-hmm.
Does that make sense? Okay. So hair evolved one time. So, but what if we take the monotremes, right? See these monotremes right over
Huh.
here? And we take
Yes.
we take this branch off, right? You see my
Uh-huh.
branch off here? We take it off and we hook it up here right next to turtles, right? And we say, okay,
Mm-hmm.
along this branch, and suddenly it branches off into monotremes right over here. Does that make sense?
What happened?
So
I
so
didn't hear
That's
you.
like
so here's
hair, so
If we
no.
break it off, but then what do we do if we break it
but,
off and then what?
but then we're gonna attach it where right next to turtles.
We would have to remove the hair part 2 then.
Right. So here's what it would be, right? So monotremes they have hair, right? We know they have hair.
Mm-hmm.
So
Yes.
here's what would happen, right? If we took monotremes and then we said, okay, they evolved from this line that's going to turtles, and suddenly they branched off here, and we have monotremes right over here, right?
Mm-hmm.
What that would mean was that hair would have had to evolve two times independently. Does that make sense, right? Because
Mm-hmm.
marsupials and placental mammals are still here, so we still have to have hair right over here because they have hair. But if monotremes have hair, well, then like somewhere over here, then hair also evolved again.
Mm-hmm.
You see how on this tree then hair would be in two different places.
Yeah, it would be where the turtles are, and then it would be in the hair thing.
Right.
I mean, where the hair already is.
Right.
Uh-huh.
So that's like a more so that's assuming that hair evolved two different times independently. So so I guess you could say that like the simplest solution, the simplest organization is usually more correct. I don't know if there's like a good analogy, but it's well, it's just that
I get it
you
though.
understand what I'm saying. So
I get what.
so if so it's more complicated to assume that hair evolved like multiple times all over the place
Mm-hmm.
versus if you just group them all together and say, okay, hair evolved once, that would make more sense, or it would be more
Mm-hmm.
more likely to occur.
But then all animals have different kinds of hair. Like sheep have like fur kind of hair, and then like bears have like what I'm
Right.
saying?
Right. But so once hair evolved, then after that, then different kinds of hair evolved.
You're saying like it branched off and then it evolved like
Right.
in different like. What you're saying is like hair came into existence like kind of. It only came into existence once, not like in a different branch.
Right.
Like it only happened once, and then all those animals came after that, and not before at
Right.
some other branch.
Right.
Right.
The idea was that if hair occurs somewhere else, that would be like a big coincidence for the exact same structure to come into being two times.
Mm-hmm.
Honest, do you understand what I'm saying?
On us.
Yeah.
Does that make sense? What we're saying that it's more. It would be like a big coincidence if hair evolved two different times, separate places on this chart.
Yes, definitely would.
Okay.
I'd confuse
All right. So
whole chart too.
so usually the cladogram is looks like this, right? So cladogram one, this is like a simple version, right? Because everything is continuing along this line, and then every once in a while something branches off and goes to the left, right? So this right over here, right, is like the simplest way of having a cladogram. So on this one, right, you see how G and H are very closely related, right? So G and H are very closely related, right? So you would say H is more closely related to G than to A, right? So, and the reason is because the common ancestor is much closer to H, whereas if you want to go back to the common ancestor of H and A, you have to go back all the way back over here somewhere. And that's where they had a common ancestor.
Mm-hmm.
Right? Now in this one, right, this is kind of here the idea is the same thing. It's just that it looks more complicated. All right, so at 1, it branches off into 2 different directions. And then over here, at 3, it branches off into 2 different directions. At 7, it branches off. When it heads over here from 3, it branches off at 6, right? When it heads over here, then at 2, it branches off, right? So in this case, right, this is the idea is the same. The core idea is the same. Is that at each point, right, some characteristic evolved and something branched off before that, right? So in this case, we would say, let's take a look at D and C, right? So we would say that, okay, D and C are closely related because they had a very, their common ancestor was not too long ago. But D versus A, you would have to go back farther in time to find a common ancestor between A and D. So you would say D is more closely related to C as compared to A. Right? Now it's also saying, let's take a look at this statement here. Clade 4 is more closely related to clade 5, right? So what is clade 4? Clade 4 is basically, A and B put together, right? There's a common ancestor right here. So the common ancestor and everything that results from it is called a clade. So this clade is closely related to clade 5. So we don't just have to look at one individual species. We could also look at clades and compare which clades are closely related together or not. All right. So we have.
Hold up, brother. Can you go back to the slide 18 one more time? All right. Can you go to slide 19?
Yeah, this one. All right. So we're going to discuss
genetic connections.
All right. So genetic connections help organize species together. The ability to see the genetic code has defined evolutionary relationships more clearly and provided evidence to support species placement. So this is kind of what we talked about before, that not just looking at, these are all like physical characteristics, right?
Right.
Looking at
so this is like genetic characteristics.
Yeah, looking at the genetic characteristics, how are we going to classify them together? So based on genetic characteristics, we're grouping, we can figure out that amphibians and reptiles, are, more closely related than before.
All right. Yeah.
So that's just like one example, right? So, all right. So, but there are some disadvantages to using genetics for categorization. So, for example, we've talked about, like bacteria. I don't know if we talked about bacteria before. So we talked about, bacteria. So bacteria, they often exchange genetic material. So let's suppose you have 2 bacteria, right? And both of them have like the same exact, sequence of DNA. So that doesn't mean that they have a common ancestor, right? So bacteria, they often, share genetic material. So I don't know if you've seen, these different kinds of ways. So there's something called transformation, right? Basically, this cell kind of releases DNA into the environment, and then this one just kind of picks it up and it implements it, right? So obviously, if a scientist comes back later and says, "Oh, well, these 2 bacteria, well, they have like very similar DNA," right? That doesn't mean that they have like a common ancestor, that's close to the present time. It could be just that this one, like this bacteria here, just released a bunch of DNA, and then this one picked it up. So you see how that makes it a little bit more confusing for some kinds of organisms, right? Transduction is when a virus takes some DNA from here and puts it into another one. And then you have conjugation, where 2 bacteria actually link up with a tube and they send DNA from one to the other.
Okay, I understand that. But can you repeat the first one again?
So this is called, so in this one, this bacteria is just releasing pieces of DNA into the surroundings.
Just like random, and then one just picks it up, kind of.
Yeah, the other just picks it up, right?
All right.
So if you see similar DNA in these 2 bacteria, that doesn't mean that necessarily they're close together, actually related close together. So this just makes it more confusing. So this is just showing a disadvantage of just looking at the genetics. All right.
Mm-hmm.
All right. So we also have non-living things. So viruses are non-living things. So you have viruses and viriyoids. All right. So what are the differences? So first of all, these are like different pictures, electron microscope pictures of different viruses. You've probably seen stuff like this. All right.
Mm.
So what is the difference between a virus and a viriyoid? So a virus is an infectious agent and has genetic material and surrounded by a protein coat. So a viriyoid is an infectious genetic particle, but it does not have a protein coat around it. So viruses and viriyoid. Now, none of these are considered to be alive, right? So these
both of these are not alive, right? Okay, yeah.
Yeah, so both of these are not considered to be alive.
But viriyoids or whatever, they're still like viruses. Are they still like harm?
Yeah,
like you can be infected with a virioid or viroid.
Right.
Right.
You can still be infected by it. Yep.
All right.
Right. This over here is kind of showing the same kind of thing, right? Viruses have a protein coat. Virioids do not have a protein coat. They don't have the protein surrounding it, but they're still infectious. Right. This over here is just kind of showing the structure of a virus. So most viruses in the middle, they have DNA or RNA. So these are kind of like fake instructions that they're going to put into cells, and then the cell, like a bacteria cell, eukaryotic cell, is going to start to make more of the virus instead of its own things, right? So surrounding that is usually like some kind of protein coat, right? So you have genetic material and a protein coat, and then some viruses, they have like kind of like a membrane around that, right? They have a membrane around that, and then other viruses, they don't have a membrane, right? So there are this virus has like an envelope membrane that's called an envelope around it.
It's not really a membrane though, right? Like you would find on a eukaryotic cell or
right. So I don't think it's as complex. So it's still made of lipids, but it's probably not going to be as complex. It's probably going to have very simple, just one or two kinds of proteins on it. That's it. Okay. All right.
So why are they considered alive?
Well, they're not considered alive because they. So these are basically almost like just particle. They don't have any. They don't do the kinds of like they by themselves cannot reproduce. They by themselves do not like use energy. They're just floating around. They're almost kind of like dead particles. But when it hits a bacteria.
So to reproduce, they basically infect bacteria and then they can reproduce, right?
Right. So they force something else to make more viruses. So they by themselves are just sitting here. They don't breathe. They don't use oxygen. They don't reproduce by themselves. They don't really do anything by themselves.
Yeah.
So these are some different kinds of shapes for the viruses. Right? And this is the same thing. You have helical shapes. This is a complex shape. This is called a bacteriophage. Looks kind of some kind of like spider, insect, or something like that. All right. So viruses, they can infect in two different ways. So when a virus infects a cell, it can infect the cell in what's called a lytic infection or a lysogenic infection. All right. So let's look at the two different types. So this one here is called lytic infection. So here's the virus, and it goes here, infects the bacteria. The bacteria makes viruses, and then when there's so many viruses in here, the bacteria just kind of explodes and just dies, and all the viruses just get released. So,
and then there's just new viruses, and then that thing keeps happening over and over.
It keeps happening over and over, right? So this.
Wait. Did I miss the lytic and lysogenic infection thing, or did you not talk about that?
So this right over here, this process where the bacteria bursts and dies, this is called lysis. So this right here is the lytic cycle. Right?
Okay, but then how does this harm the humans?
Because
it's killing off bacteria?
Because all their cells are going to be killed, right? Because like when you infect a cell, that
Oh,
cell
okay,
eventually
because this is gonna
dies.
keep happening and keep happening.
Right.
At some point, that like almost every cell is gonna be infected.
Right. So a lot of cells are going to be infected, and so a lot of cells are going
So
to be damaged and
so
die.
someone can die from this, right? Like
Right.
a human can die from this.
Yeah.
So that's why you take you take when you have like that, you don't take antibacterials, but you take like medicine that stops viruses, right?
Right. So you have
Antivirals,
antivirals.
I don't know what they're called.
Yeah.
Antivirals.
Yeah. So usually the antivirals, they don't work too well, but it's just usually most antivirals, they don't like cure the disease, they just kind of suppress it.
Mm-hmm.
So this is called this is called the lytic cycle, because this thing at the end is called lysis, when the cell bursts and all the viruses come out.
All right, I see.
Now this right here is called lysogenic. So here's what happens: the virus infects the cell, and the DNA or the genetic material goes and it gets placed inside of that cell's own genetic material, but then it kind of remains dormant. Right? So most of the time it remains dormant, or the cells can exhibit new other kinds of properties.
But okay, it remains dormant. So how does that like harm the human?
Yeah. So basically later on, some of these cells, they then kind of like the DNA kind of activates, and then that goes into the lytic cycle.
Oh, okay. So it just kind of translates back into the lytic cycle.
Yeah. So
I was thinking that it would like kind of mutate the DNA in a way or something, and then the cell would just like not be able to properly function anymore.
So there are viruses that cause cancer using this method as well, but I think that's not like a typical thing. So in this one right here, right? So when the cell is stressed or near death, the integrated DNA begins the lytic cycle. So most of the time, it's not going to produce viruses, but then at some point, some of these cells could go back into the lytic cycle and start making viruses again now. So this is how, like, some people, they're infected with something like whether it's like herpes simplex virus and stuff like that. So the virus keeps coming back again and again and again. Like, have you heard of like some people, they have cold sores, right? So the reason is like most of the time it's kind of dormant, but the DNA is still there inside those cells. And so, but then just from time to time, some of those cells start, start producing viruses again, and then again they go back dormant, and they come back later, and they go back dormant, and then some of them come back later, like that. So that's why people have like cold sores sometimes, and then it goes away, and it comes back, goes away, comes back, like that.
And it comes back.
Yeah. So that's why the lytic cycle and the lysogenic cycle are kind of like connected. So,
but you can't kill off a virus because the virus isn't alive technically, right? So how do antivirals just suppress it? So you can't get rid of a virus, or is there like a way to get rid of it?
So I think
you just have to kill all the infected cells.
Yeah. So I think the way that most of the antivirals work is so an antibiotic, right? Like for bacteria, it's actually kind of like a poison. It's kind of like a poison directly to the bacteria. But
isn't it like it purposely warms your body up because like your body heats up so that it could kill off like bacteria, right?
Yeah, I think that's,
isn't that why your body?
I think that's one theory. Like why people get like a fever is like maybe the change in the temperature could make it worse.
Is that like pseudoscience or is that actual thing?
I think that's kind of like a theory, but I mean, like as a result of any infection, like you get like a fever, but I don't know if they've. I think it's like a theory that's like that the higher temperature is worse for the bacteria.
Kills off bacteria.
But I'm not sure that's like proven or something like that.
Oh, okay.
All right. So now an antiviral usually is not like an antibiotic, like a direct poison towards the virus. Usually, what it is that an antiviral usually just tries to make your immune cells. It just tries to activate more of your immune cells, so that the body's own immune cells.
So your immune system could work it out. All right. I see.
Yeah. So your immune system could work better. Right. All right. And this kind of shows, like, on the left is the lytic cycle, On the right is the lysogenic cycle, so it just shows how they're kind of connected together, right?
Cycle.
All right. All right. That's pretty much it. There's also saying that viruses are used in gene therapy. So the idea is that, so you notice that viruses, right, can sometimes, cause DNA to be incorporated into your own DNA, right? So sometimes when a person has like a
you mean viruses can incorporate into your own DNA, right?
Yeah. So like this virus has some DNA, has some genetic material, right? And so it can
like incorporate it into your DNA in the lysogenic cycle.
Right. So the idea is that theoretically they want to use this thing called gene therapy, right? Like some people they actually have some people have genetic diseases, right? So like they actually have some kind of problem with their gene. So this idea is that maybe you could take some viruses, put in like the correct gene, and then give it to a human being, and the virus will infect all your cells with like the correct gene that will overpower the wrong gene.
But how do you know? Like it says they use an empty virus, right? So do they like inject like what's it called, synthetic DNA into the virus and then put it into your,
right.
You know what I'm saying?
Right. Correct. Yeah. So
that's what they do.
All right. Yeah. So the idea is that maybe viruses could be used as a correction method to correct the DNA of someone who has like a genetic disease, something like that. So I don't think anyone has ever like successfully done it. Like they're still they do it with, like, I think it's still just like being tested and that kind of thing. So I think it's still like in theory.
Okay.